As already mentioned, I will present today on our efforts on computation and de novo design, and as compared to Bossu's talk, which was more on the method side, I will be more on the application side in the following slides. So before starting, with the scientific work, a short introduction on large molecules research at Sanofi. So we are a global organization with sites, so one site here in the Boston area in the US, and we have 3 European sites, one in France, one in Belgium, and one in Germany. Personally, I'm based in Frankfurt at the German side, and within large molecule research, we are dealing with a zoo of different protein formats, ranging from standard antibodies, nanobodies, or bispecific fusion proteins, trispecifics, antibody, nanobody hybrids, so all kinds of different modalities and combinations of such modalities are parts of our business. So from organization-wise, in large molecule research, you saw that we have those 4 different sites, but our computational team, which is called the Digital Biologics platform, is organized as a global team, so with team members from all of the different sites. Within DBXP, we take care of all kinds of computational AI data and informatics work based on those modalities which you have seen, and we are part of the large molecule research organization. On top of that, we also have partners in our digital R&D organization. It's called the Biologics and AI short BioAIM program, where we have colleagues from global data, ML ops, infrastructure, high performance compute, which basically support our vision, which is towards biologics by design. So coming back to large molecule research, I'm overseeing the biologic AI and design teams, and within those teams we have 3 different groups. One group is on foundational models. We have a de novo protein design group and a virtual screening team, which is mainly taking care of property predictions, virtual screening applications for mono and for multispecifics. And I hope that you can see at the end of my talk that what I will present today is like you have strong inputs from all of the 3 teams, like bringing all of the different know-how, domains of expertise together for computational VHH design. Quick heads up, so I have other colleagues from Sanofi here, like Abhinav and Ui Zhang, and Abhi from the Foundational Models team will give a presentation tomorrow on our efforts on building multimodal next generation protein language models. So in case you're interested, I can highly recommend to attend this session as well. Thanks. So next, as the title suggests, it will be about nanobodies today, a quick introduction on nanobodies. So nanobodies are derived heavy chain only antibodies present in camelids. They carry a llama framework, which has 85 to 90% homology with human germlines, which we increase during sequence optimization. I mean, as the name suggests, they are single domain binding sites. They do not contain light chains or CH1 domains. So in traditional nanobody discovery, we start from immunization of llamas, and then we end up in pools, potentially NGS derived of lead molecules, where then certain leads are picked and undergo building block optimization. So as seen in the last slide on homology with the human germline, it includes framework optimization, deimmunization, PTM removal, and affinity mutations, which ultimately leads to a multi-objective optimization problem towards affinity, potency, stability, solution behavior, but also immunogenicity. So this is kind of a complex challenge which requires quite some efforts to optimize such building blocks. And basically with the de novo or computational efforts, it was a goal to augment that complex optimization procedure coming from llama-derived nanobodies with fit-for-purpose design. So the underlying idea is that we start from a fully humanized and optimized VHH framework, and then go with de novo design of the CDRs in context of a new target. So basically, like lots of the optimization efforts go fall into the domain of the framework, and basically the idea is like let's get rid of these optimization efforts and just focus on CDRs, which would significantly reduce the efforts to end up with a clinically developable molecule. So one example of such a scaffold or framework would come from the caplacizumab backbone, which is a clinically validated nanobody binding the von Willebrand factor. It's, as I said, it's clinically validated, it has good expression levels, it's highly thermostable, it shows low aggregation, and it mainly binds via the CDR loops, which you can see here. So if this kind of concept is successful, that we take the frameworks and just go for the novel design of the CDRs, basically we could keep more or less the high majority of the molecule conserved and no optimization or classical sequence optimization is needed. I mean, using ML-guided affinity or potency maturation processes might be needed in rapid cycles because, like, looking at the current state of the art, it's not likely that we would end up in kind of zero-shot CDR design already reaching therapeutically relevant affinities or even potencies. And the goal was to design an agnostic workflow which would basically be applicable to any target, other modalities like antibodies, and that the preferred nanobody framework can be exchanged. So Computational is one example, but basically generically exchangeable. So first, a bit on the considerations on what we try to consider when setting up such a workflow or toolbox. So first, we were aiming to generate a toolbox instead of a pipeline. If you look at the recent work which is published in the de novo space, it's mainly a pipeline which is kind of running in a highly conserved process. And for us, we are not competing for novelty in terms of such pipeline aspects. I mean, for us, it's about an efficient drug discovery process, which helps to solve our processes by incorporation of data which is available, but we should also be set for targets when no information is available. So what we wanted to build was a versatile toolbox for biologics de novo design covering multiple research scenarios, and it should be a plug-and-play system. So not just a straight pipeline, but that we could pull in tools, flexibly exchange tools to address specific challenges, and also have easy evaluation of new methods coming from public domain or from partners. So as I said, we are not competing for novelty, we are competing for efficiency in computational binder design, and therefore whenever there's information on binders against the target of interest, on structures, on affinities, we would like to consider this in the design process, and we would like to make use in our methods to train them, to fine-tune them on available information, because there's strong evidence that could boost the chances of success. On the other hand, the toolbox concept should also be set for targets where it's very limited or no structural, no epitope, no binding information, so that we would also be able to deal with this. But whenever more information is available, our method should be fine-tunable to make use of the available data. Last but not least, I think one goal was to make use of complementary technologies to overcome limitations of unilateral approaches and to infuse our in-house data. So one example, I mean, if we make use of AI-based approaches, heavily focusing on training data, want to complement that with physics-based approaches, sequence versus structure-based approaches, so that we look at the problem from multiple perspectives, combining many different tools, methods, and processes to filter and design our molecules. Last but not least, I think we are not there yet that it would be a fully in silico-driven process, but that we would have an optimized wet lab AI integration with very streamlined assays and rapid feedback loops to generate information which could further go and infuse next cycles of our design process. So before going into, let's say, details of the toolbox, I think we call it de novo design toolbox. But as you saw in one of the first slides, the initial goal was to keep the nanobody scaffold framework, the majority of it untouched as much as possible. So in the first proof of concept study, which we started a few years ago, which I will present today, the idea was to which extent we could keep the nanobody constant. So for the first project, which I will show today, we focused on just exchanging CDR3 or redesigning CDR3 and making use of a constant scaffold and constant CDR1 and 2 combination. Then the goal was to see how far we could go with just CDR3 design. Is it possible to reach therapeutic affinities and potencies? So as I said, that's a high-level overview of the different components we have exchangeable in the toolbox. So we have methods for epitope selection or identification. We could design starting poses, because whatever we would like to do is not unconstrained de novo design, give it a target and let the method design molecules all over the surface of the target. I think during the epitope selection, we would like to select, or we pick epitopes where we feel that by addressing these epitopes, we would reach the desired therapeutic effect. So after selecting an epitope, potentially selecting starting pose if we want to do this, then go for backbone design, structural clustering, and going back to Cosmo's talk. I mean, we also see that motifs might be generated, which we are not interested in, so we solved it via filtering removing unwanted motifs. We do ensemble generation of both targets and the binders we generate, because like proteins are not rocks, like we try to include as much dynamics as possible. Then we do sequence design. We have different scoring and evaluation schemes, which are, as I said, might be physics, energy based. We evaluate like the building block, the binder itself, but also the building block target or antigen complex. And all of this is integrated kind of flexible lab in the loop based system, where we adapt our models, we design variants, we select variants, we experimentally characterize them and run them in loops and infuse the information which we generate on the experimental data into follow-up design cycles. So the proof of concept study we started to work on a few years ago was IL-4, and we chose like several epitopes where we believed that they might be, that addressing them would result into the desired biological effect. And the data I show is on one of those epitopes we selected. So first we start with the computational design following the pipeline you saw of 3 million computational designs, and we tested 840 variants experimentally. Within those 840 variants, you can see, I mean, as I said, like we tried to keep the nanobody as much like wild type as possible. So we just changed CDR3 here, and those 840 designs included CDR length of CDR3 from 13 to 21. And you can see the BLI response on IL-4 on the x-axis. So we found 5 hits and like very low micromolar range, so from 200 to 640 micromolar. And before I will continue with the story how we went on with the next iterations of the design. Okay, first here, a bit of details on 3 of the 5 hits we found. So you can see that all of them kind of in a reasonable range of thermostability. We had also good purities, good yields, but you can see in the range of KDs they were rather far away from the therapeutic range by just designing CDR3s or redesigning CDR3s. So what we did next, we wanted to expand from there. And before I go into details of how we did that kind of hit expansion, just some information on complementary methods we are building, for example, with the foundational models team or with the virtual screening team. So in-house, we build our own protein language models, making use of our proprietary data sets of nanobody sequences. So as you can see, that suite of language models is called NanoForge. The models you see here, they were trained on roughly 300 million nanobody sequences. And basically what we were doing in the next stages of that design process, we took the binders, which we found in the first round, and we trained the model to just on the information of the known binders and the known negatives we found in testing of those 840 molecules. And we reiterated with this trained NanoForge version over the 3 million initial designs to see if by the information of the 5 hits we found and the known non-binders, we would be able to find more positive designs in the 3 million sequences. But besides that, we can do more. So the affinity model is kind of target-specific model, but we could also build target-agnostic models to filter for yield, thermostability, hydrophobicity, or polyreactivity. And on the other side, as I said, like we also can build a target-specific model, just starting from the hits we have and use the LMMs to design mutations which might help better to accommodate the de novo design CDRs, which could boost stability but also affinity, and we can train like binding affinity models to discover new hits. And basically, this is what we did, and here's a bit more information on where we go with our models. So like as I said, we started with 300 million nanobody sequences, and in 2025 there was a big investment into tripling down on the nanobody sequence data to train the PLMs, and we are with 1 billion sequences now. We explore in the foundational models team different architectures, so it includes BERT models, T5, mixed-RAL GPT models. Like as you can see or might know, they have different application domains. So BERT is an encoder-only model which is strong in downstream prediction tasks. We also have like the decoder only, which are better in painting and generative tasks. And also we have versions with different levels of parameters ranging from 20 to 300 million parameters. And some of the learnings along those training and benchmarking processes is that it makes sense to train on non-redundant sequences. So it's not just the pure amount of nanobody sequences, it's about the diversity. And like defining the diversity is, the best way is not to go on sequence identity only. There are smarter ways to do so. And bigger models in terms of parameters do not like equal better performance in downstream applications. So this is just some examples. So for example, I mean, as I said, like this is what I try to visualize here. I mean, we keep the frameworks constant, and we only exchange CDRs. So we generated a custom data set which includes several nanobody frameworks which are clinically validated and where we believe those are strong and optimized frameworks. We generated diversity in CDRs, used a mammalian expression system and generated more than 4,000 clones, tested it for yield, and then built kind of a nanoforge-paired downstream application to predict the yield, which we can then use like for the de novo designs to filter out molecules which are predicted to have low yields. We do similar things for other properties. Second slide is not changing, there we go. For example, on thermal stability, so again we generated more than 4,000 nanobody variants for in conserved frameworks which we use for de novo design and exchange CDR diversity. We have more thermostability data points, but those data points come from different frameworks, and we wanted to generate specific data, boosting our models for nanobody de novo design, where we keep frameworks constant, no mutations in the frameworks already optimized, and just like exploit CDR3 diversity. So again, you can see that we also end up in reasonable models for nanobody thermostability prediction, and we also look at polyreactivity. But we see that pure polyreactivity alone is maybe not best predictive of, let's say, unspecific binding or solution stability. That's why we perform polyreactivity assays after stress. So we take a sample, we perform polyreactivity assays, then we stress it by thermal stress, and then we basically generate the delta in polyreactivity increase after stress, and this we consider as more reliable result than, let's say, unstressed polyreactivity measurements. And all of those models, like the affinity model as well as the property models, are then used to guide the next steps, or also with the target agnostic models, the initial steps of our de novo campaigns to not only get binders, but already filter out everything with low expression, low TM, and so on and so forth. So what we did here, like just a quick recap. So in the first stage, we tested 840 molecules covering CDR range from CDR range 13 to 21. We found 5 hits in the very high micromolar range. We used those 5 hits and the 835 negatives to train an affinity PLM model based on our nanobird models. And then we just filtered the 3 million design, or the part of the 3 million designs which includes CDR range 13, because we found most of the hits, as you can see on the top left side, in CDR range 13 and 14. So we first thought, okay, let's see, can we expand hits like in that shorter CDR range? So we tested 210 compounds and we found 7 additional hits. So then we went to the next stage and took the total of 13 hits we found, and again, like, retrained the affinity classification model, and with then a total of 12 hits. And then we filtered, but again, for the overall CDR distribution of 13 to 21. And you can see with those 30 hits, training that affinity classification model, we just tested another 280 variants, and now we were able to find hits all over the place, with the exception of CDR range 17. We could find very diverse panel of hits binding to IL-4 in the single-digit micromolar range. So then we moved on from this, and we wanted to see how far we could push the approach to more therapeutically relevant affinities. But before, you can see here an overview on the biophysical properties. On the left side, you can see an overview on TM. So in blue, you see, in orange, you see the non-binding molecules. In blue, you see the binding ones, which we selected. And specifically for the binding ones, you can see that the majority is above 60 degrees. Then the next plot is on the polyreactivity assay. Most of the compounds show very low polyreactivity in terms of aggregation. Also, specifically, the hits are highly pure, and in terms of yield, we also have very reasonable nanobody expression rates. So it shows that kind of the design approach works, but also that the filtering which we applied made sense. So then in the next steps, we wanted to push the affinities, or to see how far we could go, and if we could reach therapeutically relevant affinities by, let's say, kind of this pure de novo identified hits from first stage. So together with our partner DeepTope, we performed deep mutational scanning from 3 of the initial hits. So the first 3 basically, which I showed in the first hit identification stage. So before we went for the hit expansion, we did like single point deep mutational scanning, and then we selected 2 of those hits for recombinatorial screening. And the results you can see here. So we went with 1 VHH with CDR length of 21, and we were able to find 8 clones in the range of 5 to 50 nanomolar. And for the VHH, the second VHH we chose, that was one with shorter CDRs of 13 amino acids, we found 6 clones in the range of 1 to 5 nanomolar with no biological polyreactivity reagents and highly stable TM via yeast display approach. So then we went for upscaling of those hits. Not moving. Can you maybe change the slide for me, because here it's not moving anymore? There, okay, perfect, thanks. So after upscaling, out of these, I think 13 hits in total, 8 of them, so like the deep mutational scanning and combinatorial approach was a yeast-based approach. We then went for upscaling, and out of those 13 hits, 8 of them showed very good biophysical properties in terms of production, TM, aggregation, and you can see that via SPR, we could even confirm that the hits are in the picomolar range, so ranging from 260 picomolar to 11 nanomolar. I don't have the data on the slides yet, but the compounds were also tested in reporter assays to show if they would be antagonists in the IL-4 pathway, and the reporter assay confirmed that I think most of them are in the picomolar range with antagonistic activity against IL-4, which also shows that the epitope selection kind of worked out well. Also, in terms of the epitope selections, we don't have crystallographic structures yet. This is still ongoing, but we performed mutational studies knocking out certain residues from our models, which we believe are the main drivers of the activity. And by knocking out those amino acids, we completely kill binding against the target. Before going into the lab into the loop concept, maybe one further consideration for the last slide. So all of the mutations which were introduced to the wild type hits coming from the de novo design, so the CD3 designs, the mutations found via the yeast display approach all were in CDR1 and 2. So basically we left CDR3 untouched, and we get those huge shift in affinities by modifying CDR1 and 2. This is why our further iterations of the de novo pipeline also design all of the 3 CDRs kind of in parallel. And the number of mutations was in the range of 6 to 8 additional mutations which we needed to incorporate in CDR1 to 2 to come from the, let's say, single-digit micromolar to the single-digit nanomolar or picomolar range. So all of this would not be possible without a very close lab-in-the-loop concept integration. So basically we pushed or further customized the lab, to be the perfect partner for the computational approaches, and we go for production in mammalian expression systems. Also perform the purification, binding via BLI and SPR. We perform ASEC and STS for aggregation, thermostability, counter screens on related targets, or also on the initial target of the nanobody building blocks we use for the, or backbones we use for the de novo design, polyreactivity and functional assays, which we can all do in up to 7 weeks and get the full package to have really fast iterations in that design process. You can see a picture from the lab there, so it's like really a huge suite of automation which is constantly adapted and new assay technologies are included. And there's one example on the assay side. So as you saw, we are working in first place with very weak binders, and therefore we needed to adapt the technology to be able to pick up those weak binders. So some of the colleagues say, all you need is a hit, because if we have a hit, then we can expand via the language models and like get many more hits, but you need to be able to find such hits, even if they are very weak. So our colleagues in the lab developed a BLI screening assay with an anti-MYC antibody pre-incubation, so that we could kind of extend the size of the complex to pick up really weak hits. And what you can see there, basically the baseline is without any anti-MYC pre-incubation, so we get no binding signal. But if we do the anti-MYC antibody pre-incubation, you can see that you end up in kind of valid binding curves that we can pick low affinity hits, which are very informative for our approaches for diversification, but also affinity optimization. And with that, I would like to summarize. So we developed a versatile, or the goal was to develop a very versatile toolbox for nanobody de novo design, which can cover scenarios with no information on epitopes, structures, binders, sequences, but also be set for, or can make use of any available information as well. I mean, we aim for the close integration with the wet lab, and we had state-of-the-art hit rates of more than 100 hits in just 1,300 screen variants, just by designing CDR3. And like the evidence we have now, like we are applying co-design of all 3 CDRs, but the goal is to keep CDR1 and 2 as untouched as possible, so we just change like small strings there and could directly push by these affinity levels. We leverage tools, complementary tools for AI-based hit expansion, diversification, and hit optimization, and generally we are able to get favorable developability properties. And we could show here that by starting with CDR3 design, I think identified hits can be optimized to reach therapeutically relevant affinities and also potencies in reporter assay setups. And also, like, we see a wide diversity, so you saw we could cover from CDR range 13 to 21, and also the sequences are highly diverse, and we have, like, let's say plenty of diversity to choose from. So we have lots of internal partners, like lots of the work was